Hello, this is Jim Schnabel giving the fourth talk in a series from the seventh edition of Principles of Biochemistry, authored originally by Albert Lehninger. Today we're going to be talking about evolutionary foundations, which talk about the information storage, the molecules that have been evolutionarily produced to create fidelity through generations of various living things. And we will be talking about RNA and DNA and the interaction that they have to produce protein and maybe some fundamental definitions. So we'll be talking about this when we get to nucleic acid structure, but the nucleic acids have bases, and there are four main bases in DNA and four main bases in RNA with a switch of one for the other. So we have complementary strands, so adenine, which is a pyrimidine, a single ring, pairs with thymidine. I'm sorry, adenine is double ring, pairing with thymidine, which is a single ring, guanine double ring, pairing with cytosine in DNA, and uracil replaces thymidine in RNA. And these are hydrogen bonds, and they have remarkable fidelity with a very low error rate that particularly is important as you're young. The errors increase with age, and this is due to several different possibilities. We refer to a DNA template. DNA in an organism can either exist as a single strand of DNA or a double strand of DNA, and the double strand is evolutionarily more advanced and is another level of structural fidelity and reproduction fidelity in the organism. But the template is a single strand, and from that DNA template single strand you code and get what's called an RNA transcript, which is also a single strand. This RNA transcript is used for protein synthesis. We talk about translation, which is the production of protein from the RNA single strand transcript. From an evolutionary standpoint, it is probable that RNA began first, and the presence of reverse transcriptases illustrates that there is the capacity for RNA segments to be transcribed, reverse transcriptase back transcribed to DNA template, and then from that DNA template it can produce RNA copies. A lot of viruses with reverse transcriptase, with human immunodeficiency virus being the predominant example, is an example of that. The protein is synthesized as a single strand, and as the protein is synthesized, it will fold spontaneously into a three-dimensional native conformation. In some cases, the protein is a monomer unit. There are multiple units. They can be 2, 3, 4, and those will associate with each other through a variety of different means, usually ionic or coordination bonds. And you get more complex structures of proteins can bond with carbohydrates. They can react in all sorts of myriad ways. And again, the microenvironment, pH of the solution, metal ions, can affect the native conformations. You get perturbation of the structure and a greater propensity to mutations if you have structures that will cause issues with the structure, whether the structure is a protein or a RNA template, or more critically, DNA, if the organism is using DNA as the means for transcribing more and more copies of itself. From an evolutionary standpoint, these biomolecules began from simpler forms, probably, and there were classic experiments done back in the day by Stanley Miller in 1953, where electricity was generated through a flask with simple molecules, ammonia, methane, hydrogen gas, water, hydrogen sulfide, and you could get some simple amino acids and hydrocyanide from that mix. And it's thought that these, with enough time and energy and position, you can get more and more complicated molecules, and at some point, these molecules were able to replicate. So when we talk about mutations, a lot of mutations are silent. You have them, but you don't know them. Some mutations are beneficial. They provide or construe an evolutionary advantage over wild-type unmutated cells, or they can be lethal mutations. Some are point mutations. One nucleic acid changes, and you have a deleterious outcome. Oftentimes, these mutations are multifactorial. They occur at different points in the genome, and you end up getting problems with particularly in areas such as carcinogenicity and risk for proliferative diseases, or in the case of deficiencies, no substance produced at all, and you build up large amounts of the non-metabolizable form. So DNA is, again, as I said, probably occurred later in evolution, and this was probably DNA probably occurred later on to produce a more stable informational system, to the point where eukaryotes actually have a second membrane with strict permeability requirements inside the cell where the DNA is placed. And prokaryotes will have single-strand plasmid DNAs, and that would be a different process, and those are more prone to a genetic alteration. So that's it for the talk, and thank you very much for listening.